Students, till now we have seen the application of biotechnology in agriculture. We have seen two examples. One is production of BT cotton, another one is production of pest resistant plants, that is parasite nematode resistant plants. And we have some more examples which are having application in agriculture, which is not mentioned in the textbook, but they are the competitive point of view. I am going to give you another two more examples for application of biotechnology in agriculture, right? So one of the advantages of the GM crops, if you see, there is one advantage: enhanced or enriched nutritional value of food. Is it is one of the advantage of GM crops, that is enhanced or enriched value of food, and another one is reduced post harvest losses. Right, these are the another two advantages of the GM crops. So in the reduced post harvest losses, I have explained with an example, right? It was production of flavor savor tomatoes. These tomatoes are genetically modified tomatoes. So this is also the product of biotechnology, that is the production of flavor savor tomatoes. Now what are these flavor savor tomatoes? These are the genetically modified tomatoes, and these are the first transgenic crops to reach to the market in America. In India, BT cotton is the first transgenic crop to reach to market, whereas in case of America, flavor savor tomatoes were the first transgenic crops to reach market. The flavor savor tomatoes or the transgenic tomatoes, why they are so important, or what are the advantages they are possessing? So what happens in the normal tomatoes? In the normal tomatoes, after the harvesting, they are transported to the market, okay, wherein the end user, the customers, buys the tomatoes, right? If you see these tomatoes which we take, okay, they get ripened soon. So what is the reason behind the ripening of the tomatoes, right? And in the first year you people have learnt in biomolecules chapter, what is the cell wall composition of the plants? Plant cell wall contains, or it is made up of, cellulose, hemicellulose, then pectin and some of the minerals. Now pectin is once again contains galactose. Pectin is made up of galactose. So there is an enzyme known as polygalactouronase enzyme is produced, which is going to, polygalactouronase enzyme is going to cleave the cell wall of the plant cell, right? So polygalactouronase cleaves the pectin content. This is an enzyme which is going to degrade the pectin in the cell walls and making the fruit softer, which is going to make it susceptible for the fungal infections, right? That is the reason these tomatoes get damaged soon while transporting until it reaches the end user. So in order to maintain the stability as well as firmness and to save the flavor of these tomatoes, biotechnology has developed a technique to delay in ripening of these tomatoes, to delay in the fruit ripening of these flavor savor tomatoes. That is by blocking the gene coding for polygalactouronase enzyme. If the gene is blocked for the polygalactouronase enzyme, what will happen? Then the gene coding for polygalactouronase enzyme will not be expressed. If the gene is not expressed, the protein is not formed. If the protein is not formed, then there won't be the enzymatic action, because enzymes are proteins. Yes or no? Enzymes are proteins. All enzymes are proteins, but not all proteins are not enzymes, because some proteins are hormones, some proteins are enzymes. Clear, students? So there won't be the formation of the protein and there won't be enzymatic action taking place. So this is how the flavor saver tomatoes were developed, by blocking the native gene that is coding for the enzyme polygalacturonase. So therefore production of these flavor saver tomatoes remained fresh and firm with delay ripening till it reached the end users. If you see the normal wild type of tomatoes, they or the organic tomatoes, they get ripened soon and whose cell wall is very much susceptible for the bacterial and fungal infections. They get damaged soon during transportation. Why? The cell wall gets degraded. Once the cell wall is getting degraded, the inner fruit gets exposed to the microbial action and therefore the fruit gets damaged and results in the spoilage. So to prevent that, the biotechnology technique that has developed is blocking the gene coding for polygalacturonase, which developed the firm and fresh tomatoes with delay ripening and saving the flavor of these tomatoes till it reached the end users. Another example of application of biotechnology in agriculture is production of golden rice. Production of golden rice. What is this golden rice? The rice which appears gold in color. Is it, students? This is the transgenic rice. Transgenic rice, which is known as golden rice. What happens in this transgenic rice? The transgenic rice or the transgenic Oryza sativa, which is the scientific name of rice produced, is having yellowish in color. This rice is having yellowish in color, so that's the reason it has got the name golden rice. It has got the name golden rice. What is the speciality about this rice when compared with the normal rice? Okay, it has got the high content of precursor beta carotene. Beta carotene is one of the precursor in the synthesis of vitamin A. Everybody needs vitamins, right? So in the biosynthetic pathway, if we see in the metabolic pathway in the synthesis of vitamin A, beta carotene acts as a precursor for the synthesis of vitamin A. Now this vitamin A, why it is important? Because the deficiency of vitamin A leads to the night blindness. In many countries, the children are suffering from night blindness. So in order to prevent that, the rice itself is getting enriched with the precursor beta carotene. So the rice plants were transformed by incorporating certain genes which alter the metabolic pathway of carotenoid synthesis to produce high content of beta carotene. In the plants, in the rice plant, beta carotene is produced, but using this biotechnology, the beta carotene is produced in the edible part of the plant. It is being produced with a high content in the edible part of the plant. So the rice plants were transformed by incorporating 3 genes, which are PSY, that is coding for an enzyme known as phytoene synthase. Phytoene synthase. The gene that is PSY, that is isolated from daffodil plant. It is isolated from daffodil plant and it codes for phytoene synthase. Another gene, that is CRTL. Another gene is CRTL, that is isolated from a bacteria Erwinia uridovora. It is a soil bacteria. From this bacteria, the CRTL gene is isolated and that codes for an enzyme phytoene desaturase. It codes for an enzyme phytoene desaturase. There is another gene LCY, that is coding for lycopene cyclase enzyme, and it is also isolated from daffodil plant. There are three enzymes. These are introduced in the transgenic plants. These are introduced in the rice plants, okay, and it is introduced in such a way that they are under the control of endosperm specific promoter. Means in the, if you see study in the molecular basis chapter, the expression of gene, see in which location the gene has to be expressed. Now the proteins are produced in the body, but some proteins are produced in each part of the body. Different types of proteins are formed. This is all controlled by the gene expression system. In that, a gene known as a promoter gene. Right? Now, so this gene is placed under endosperm specific promoter, so that this particular beta carotene will be expressed in only endosperm. Getting students? In only endosperm, that is in the edible part of the plant. So therefore, this is how the golden rice is produced. So using the genetic engineering technique, the genes, that is PSY, LCY and CRTL, are cloned into TI plasmid of agrobacterium and transferred into rice nuclear genome, that is into the plastids, and it is expressed in the endosperm because they are placed under endosperm specific promoter gene. So then it is going to produce the beta carotene in the edible part of the plant so that it can be utilized. And these three enzymes are playing an important role in the metabolic pathway or the synthesis of vitamin A. So each enzyme has got its catalytic activity in the conversion of one biomolecule product into the another product. This is how the production of golden rice is going to takes place. Let us see now competitive bits with respect to the concepts which I have discussed, that is BT cotton, BT parasite nematode resistant plants, that in the tobacco plants, and flavor savor tomatoes and production of golden rice. Okay, question number 1. The main technique involved in agricultural biotechnology is called A. Tissue culture, B. Transformation, C. Plant breeding, D. DNA replication. Which is the main technique involved in agricultural biotechnology? Now in the agricultural biotechnology we have seen, okay, that is in case of the BT cotton also, in if you are using a gene gun method or agrobacterium mediated transformation method, what we are going to, we are introducing the desired genes in the TI plasmid, and then what we are doing, we are introducing this TI plasmid in certain plant cells. Means some part of the plant we are taking and introducing this, and externally in the artificial nutrient medium we are developing that small part of the plant into the whole plant, which is known as regeneration of the plant. Okay, that process is known as tissue culture. So option A is the right answer, that is a tissue culture is the right answer. Moving on to the question number 2. Consumption of which one of the following foods can prevent the kind of blindness associated with vitamin A deficiency? Consumption of which one of the following foods can prevent the kind of blindness associated with vitamin A deficiency? Option A, BT soybean. Option B, golden rice. Option C, flavor savor tomatoes. Option D, BT cotton. So, golden rice, option B is the right answer. Option B, golden rice is the right answer because golden rice is rich in beta carotene, which is a precursor for vitamin A synthesis. So, question number 2, option B is the right answer. Moving on to the next question. Question number 3. Which of the following is false for BT transgenic plant? Bt transgenic plant. Option A, it is disease resistance. Option B, prepared by Bacillus thuringiensis. Option C, it is recombinant type. Option D, no such plant is known, which is false about Bt transgenic plant. Yes, Bt transgenic plant is disease resistance because it is having a resistance towards the insects, so no more the insects can damage the plant. And option B, prepared by Bacillus thuringiensis, yes, it contains the genes that are isolated from Bacillus thuringiensis known as cry genes. Option C, it is recombinant type. Yes, transgenic plant is developed by recombinant DNA technology. So A, B, C are true for Bt transgenic plant, and option D, no such plant is known, is right option, which is false about Bt transgenic plant. If you see question number 4, tobacco plants resistant to a nematode have been developed by the introduction of DNA that produced in the host cells. Tobacco plants resistant to nematode have been developed by the introduction of DNA that produced in the host cells. Option A, both sense RNA and antisense RNA. Option B, a particular hormone. Option C, a vitamin. Option D, a toxic protein. So we are introducing the DNA, that is, or the genes specific to the parasite nematode in the plants in such a way that it is going to generate both sense RNA and antisense RNA, because the sense RNA and antisense RNA, since they are complementary to each other, they are going to form the double-stranded RNA, and double-stranded RNA is going to initiate the RNA interference pathway. So RNA interference pathway is going to destroy the specific mRNA or the target mRNA of the parasite, which is going to remain non-functional and is going to destroy the parasite nematode. So option A is the right answer for question number 4. Moving on to next question, that is question number 5. Bt cotton is not a GM plant. It is not insect resistant, or it is not a bacterial gene expressing system. Option D, it is not resistant to all pesticides, which is the right option. Let us see one by one. Bt cotton is not a GM plant. No, it is false. It is a genetically modified plant. So, okay, Bt cotton is not insect resistant. No, it is having resistance towards a group of insects. And option C, it is not a bacterial gene expressing system. Yes, it is a bacterial gene expressing system because it is going to express the genes that are isolated from the bacteria Bacillus thuringiensis. So, and Bt cotton is not resistant to all pesticides. Yes, it is not resistant to all pesticides because in case of Bt cotton, it contains the genes cry1AC and cry2AB, which are resistant to cotton bollworms only, not to other insects. Getting? Because these Bt toxins are highly specific based upon the crop and based upon the plant, it is going to be isolated and expressed. So option D for question number 5. Option D is the right answer. Option D is the right answer. Question number 6. In RNA interference, genes are silenced using single-stranded DNA. Option B, double-stranded DNA. Option C, double-stranded RNA. Option D, single-stranded RNA. In RNA interference, what is required? Complementary double-stranded RNA is required, which is going to initiate the RNA interference pathway. So option C, that is double-stranded RNA, is the right answer. Clear? Moving on to next question, that is question number 7. Activation of toxin of Bacillus thuringiensis is due to option A, acidic pH of stomach, high temperature, option C, alkaline pH of gut, option D, mechanical action in the insect gut. I repeat, activation of toxin of Bacillus thuringiensis is due to acidic pH of stomach, high temperature, alkaline pH of gut, mechanical action in the insect gut. Yes, the Bt toxins are produced initially as protoxins or inactive toxins which gets activated in the gut of the insect. It is because of the alkaline pH of the gut of the insect that is going to solubilize the crystal protein and is going to activate the toxin. So, question number 7, option C is the right answer. Alkaline pH of the gut is the right answer. Moving on to question number 8. Cry genes in Bacillus thuringiensis code for. Cry genes in Bacillus thuringiensis code for option A, Bt toxin, option B, normal proteins, option C, vitamins, option D, lipids. Option A, Bt toxin. Cry genes in Bacillus thuringiensis code for, it codes for delta endotoxin, or it codes for crystal proteins, or it codes for Bt toxin. So option A is the right answer. Instead of option A, if they give crystal protein, also right answer. If they give delta endotoxin, also right answer. Or if they tell cry protein, then also it is a right answer. So option A is the right answer for question number 8. Clear, students? Now moving on to question number 9. Bt toxins or cry proteins are released in which growth phase of organism? Yes, when I was explaining about the Bt toxins, so I have already explained like in which growth phase these Bt toxins are produced, or these genes, cry genes, are expressed in which growth phase. Option A, log phase, option B, lag phase, option C, death phase, option D, sporulation. So that is option D, during spore formation the Bt toxins are produced. Question number 9, option D is the right answer. Then moving on to question number 10. Examples of Bt plants are option A, cotton and corn, option B, rice and potato, option C, tomato and soybean, option D, all of these. Yes, the examples of Bt plants or the Bt toxin genes are introduced to produce the insect resistant plants include cotton, Bt cotton, Bt corn, rice, potato, tomato and soybean. So option D, all of these are Bt plants. So option D is the right answer for question number 10. Dear students, now moving on to the next question. Question number 11. Which of the following genes were introduced in cotton plant to protect it from cotton bollworms? Which of the following genes were introduced in cotton plant to protect it from cotton bollworms? Option A, CRY-AC and CRY-AB. Option B, BT-AC and BT-AB. Option C, CRY-1AC and CRY-2AB. Option D, NIF genes. So, the Bt cotton plant is developed by inserting genes CRY-1AC and CRY-2AB which produce the Bt toxin that are effective against the cotton bollworms. So, option C is the right answer for question number 11. Then question number 12. Which of the following is the nematode that attacks the roots of tobacco plants? Which of the following is the nematode that attacks the roots of tobacco plants? Option A, Agrobacterium tumefacius. Option B, Rhizobium leguminosarum. Option C, Melidogene ecognitia. Option D, Tinea solium. So among these, the one which belongs to the nematodes is the species Meloidogene incognitia, that is going to attack the roots of the tobacco plants and cause the disease root knot disease and damage the tobacco plant, which is a parasite nematode. So option C for question number 12 is the right answer. Option C is the right answer for this question. Then moving on to question number 13. Bt toxin kills insects by option A, inhibiting protein synthesis, option B, generating excessive heat, option C, creating pores in the midgut cells, cell swelling and lysis, option D, obstructing a biosynthetic pathway. Bt toxin kills insects by which mechanism? That is the question. Is it going to inhibit protein synthesis? No, the protein synthesis inhibition is taking place in RNA interference. Then it is going to generate excessive heat? No. Option C, creating pores in the midgut cells? Yes. Once the protoxin is getting activated to toxin in the gut of the insect, what happens? It is going to create the pores and it is going to fill the cells with a fluid leading to cell swelling, and once the cells are swollen they get lysed and the toxic content is released to the outside the neighboring cells to other cells and eventually leading to the death of the insect. So option C is the right answer for question number 13. Question number 14. Bt corn has been made resistant from corn borer disease by introduction of the gene option A cry1AB, option B cry2AB, option C ampicillin resistance gene, and option D TRP gene. Yes, Bt corn has been. The Bt cotton is developing resistance by introduction of the genes cry1AC and cry2AB. So Bt corn has been made resistance from corn borer disease by introduction of the gene cry1AB. So option A is the right answer for this question. Cry1AB is the right answer. Then moving on to the last question, that is question number 15. First genetically modified plant commercially released in India is. First genetically modified plant commercially released in India is golden rice, flavr savr, Bt brinjal, Bt cotton. So don't get confused in this question. They are asking first commercially released in India, that is Bt cotton. Flavr savr is first released in the market in America, but in India it is Bt cotton. So option D is the right answer for this question. Dear students, we will continue in the next class.